There is a genetic signal in a pyramid in Guanajuato that does not match anything. Not anything in Mexico, not anything in North America, not anything in the entire Western Hemisphere, not in any ancient genome ever sequenced, not in any living population ever sampled. It came back from the sequencer and the scientists looked at it and the only word that fits is impossible. The site is called Cañada de la Virgen. It sits in the high desert of central Mexico, about 250 kilometers north of Mexico City. People built there, lived there, buried their dead there, continuously from around 540 CE until a megadrought finally emptied the place sometime around 1050 CE. Archaeologists have been cataloging the bones for years. In 2017, a genomics team arrived with different equipment. Not brushes, not measuring tape. Extraction kits, one tooth, one femur fragment, one result that has no source. Viridiana Villa-Islas leads the ancient DNA lab at the International Laboratory for Human Genome Research at UNAM, the National Autonomous University of Mexico in Querétaro. She and her colleagues spent years building the most comprehensive ancient Mexican genome dataset ever assembled. 12 individuals, 27 mitochondrial sequences, 8 archaeological sites, spanning 830 years of pre-Hispanic history. When the Cañada de la Virgen data came back, they found what they named UpopA2, a ghost population, a lineage contributing roughly 17% of the ancestry of ancient individuals buried at that site, with no identifiable source population anywhere in the Americas, past or present. Everything I just told you, hold on to it, because it's about to mean something completely different. Think about what that actually means. Every major ancient DNA study of the Americas over the past 15 years has been building toward a complete picture of who moved where and when. The database is enormous, and into that enormous database, a signal appears that matches nothing in it. Not a gap in the data, not a sampling error, a people, a real population of human beings who lived, moved, reproduced, left their DNA in the bones of the people buried under a pyramid in Guanajuato, and then vanished so completely that their closest genetic relatives, if they exist anywhere on Earth, have never been found. If you've ever actually wondered where you come from, not the family story, not the national story, the real biological answer, you're going to want to subscribe. Because what this research is showing about Mexican DNA goes deeper than anyone expected, and it starts with a name that no one has. The same ghost, a related but distinct signal, designated UpopA, appears in a completely different region of Mexico, in a completely different archaeological context over 1,000 kilometers away. 2 signals, 2 sites, no explanation, not one. 1,000 kilometers north, the Sierra Tarahumara rises above 2,400 meters in the state of Chihuahua. The Rarámuri people, most of the world calls them Tarahumara, have lived in these mountains since before any European arrived on this continent. Some communities maintain a degree of isolation that has lasted essentially into the present day. And in the ancient genome of an individual buried there, the Villa-Islas team found UpopA, not UpopA2, but the original signal, contributing approximately 28% of that individual's ancestry. 2 sites, 2 ghost lineages, no known source for either one. But that is not the finding that stops you. The finding that stops you is what Dr. Andres Moreno Estrada published in 2014. Moreno Estrada is based at INMEGEN, Mexico's National Institute of Genomic Medicine, and his team spent years building something that had never existed, a genome-wide dataset of Mexican indigenous populations at real scale, over 1,000 individuals, 20 indigenous groups, 11 mestizo populations, the most comprehensive picture of living Mexican genomic diversity ever assembled. And what it showed broke the frame of the standard model completely. Some indigenous Mexican populations, separated by as little as 400 kilometers, are as genetically distant from each other as Europeans are from East Asians. Let that sit for 1 second. That is the deepest level of divergence that exists between any 2 human populations anywhere on Earth. The genetic distance between a person from Norway and a person from Japan. Between groups living in the same country, sometimes within a day's drive of each other. Here's where it gets strange, and I mean structurally strange, not just surprising. The standard model of Mexican identity runs like this. There were indigenous peoples, then the Spanish arrived, then the mixing happened, and what emerged is the mestizo, a new population carrying all 3 streams. That model treats the indigenous component as a unified base. One thing, a starting point. What Moreno Estrada's data proves is that the indigenous component is not one thing. It never was. The populations that predate the colonial encounter are as genetically distinct from each other as the colonizers were from them. The word indigenous as a genetic category is a political boundary drawn over a biological chasm. And if that's true and the data says it is, then 65% indigenous ancestry on a consumer DNA test is not a statement about one people. It is an average across populations as divergent as continents. Test has answered a question while hiding a more important one. Here's what the ancient DNA established next. The assumption that a catastrophic megadrought in the 10th century caused a mass population replacement across central Mexico. Hunter-gatherers from the arid north sweeping in to fill the vacuum left by collapsing Mesoamerican civilizations was consensus archaeology for decades. The DNA says the opposite happened. What actually happened, according to the bones, is nothing. Around 900 CE, the northern frontier of Mesoamerica begins to shift. The boundary between the agricultural civilizations to the south and the semi-arid nomadic territories to the north starts moving southward. Archaeological evidence shows sites abandoned, trade routes collapsing, political centers losing population. The drought lasts roughly 2 centuries, 200 years of documented climate catastrophe across central Mexico. And the people at the border? The populations caught between two worlds in the zone where Mesoamerica bleeds into what archaeologists call Aridoamerica. They didn't leave. They weren't replaced. The ancient genome data from sites before the drought and sites after the drought belong to the same continuous population. The Villa-Islas study confirmed it. The genetic distance between the pre-drought individuals and the post-drought individuals from the same frontier region is statistically zero. This discovery was made public in 2023. It has barely been discussed outside specialist circles until now. The textbook said there was a replacement. The bones say there was continuity. Those are not two interpretations of the same data. They are contradictory findings, and the bones win. The team's explanation for how they survived where the models predicted they couldn't: a mining-based economy. The agricultural civilizations to the south failed when the rains stopped. The frontier populations had another industry that didn't depend on rainfall. The economy saved the genome. The genome remembered what the archaeology forgot. Think about what that means for everything else we've assumed about pre-Hispanic Mexico. The standard model was built on archaeology, on what was found in the ground, what was abandoned, what burned. Ancient DNA is now running that model against a biological record that does not interpret, does not editorialize, does not write history. It simply says who was there. And again, the answer is not who we thought. 900 years after that drought ended, the descendants of the people who stayed are still in the Sierra Gorda, the same families, the same lineages, carrying a genome that 2 millennia of archaeology had, in a meaningful sense, gotten wrong. But the survival story of central Mexico gets more complex still. Because the populations who endured the drought, who outlasted the collapse of named civilizations, they are also carrying traces of a founding event so severe that it nearly ended the entire human story of the Americas before it began. The math of survival in the ancient Mexican genome runs in one direction only. And what that direction reveals changes how every percentage on every ancestry test should be understood. The survival math changes when you understand the scale of what it was surviving. Mexico City, 2023. A DNA ancestry test arrives in the mail. The result reads 62% Indigenous American, 33% Iberian, 5% West African. The person who opens it is from Guerrero. They post it online. The comments say, that sounds about right. And in one sense, it is about right. In another sense, the genomic sense, it is hiding more than it is showing. Dr. Humberto Garcia Ortiz and colleagues at the Instituto Nacional de Medicina Genómica analyzed genomic data from 716 indigenous individuals across 60 ethnic groups in Mexico. 72 communities, the most granular survey of indigenous Mexican genomic diversity ever conducted. And what it revealed is that the variation within the indigenous component of Mexican DNA is not just significant. It is continental in scale, and it extends beyond Mexico's borders entirely. Some individuals in the dataset showed Mayan ancestry in regions far from the Yucatan. One showed indigenous American ancestry tracing to Bolivia and Peru. The genetic structure of Mexico is not contained within Mexico. Stay with me, because this next part is the reason this video exists. The 62% indigenous American number on that ancestry test was calculated against a reference panel. A reference panel is a database of genomes from known living populations. It's the ruler against which your DNA is measured. The problem is what's not in the ruler. The ghost populations found in the Villa-Islas 2023 data, UpopA and UpopA2, were not sampled when those reference panels were built. They weren't known. They couldn't have been included, which means the results coming back from consumer ancestry tests for people of Mexican descent are systematically incomplete in a way that cannot be fixed by taking the test again with a different company. There is ancestry in living Mexicans that no commercial test currently measures. Not because the test is wrong, because the reference population it measures against is missing people who left no other record. And that matters most when you learn where the ghost signal actually lives. It doesn't only appear in bones in a pyramid. The Villa-Islas team found UpopA, the original ghost lineage, in a living population. Today, right now, the Mixe people of Oaxaca carry this signal in their living genomes, a lineage from a population with no known origin, no name, no surviving culture, present in the DNA of a living indigenous community in southern Mexico, who have been carrying it for thousands of years. They have been carrying a ghost, and the ghost is not from anywhere we have found. Thousands of years before the Ancestry test exists, a different kind of arrival is happening on the coast of what will become Mexico. Veracruz, 1519, the Spanish land. Within years, some historians argue within months of permanent settlement, the first enslaved Africans arrive in New Spain. By 1650, more Africans than Spaniards live in the territory. This is not a footnote to the colonial story. This is the colonial story. And yet it is the component of Mexican DNA that the standard narrative, the textbooks, the national identity, the census categories that existed before 2015 handled least. The average Mexican carries approximately 4 to 5% West African ancestry in their autosomal genome. That number sits low and relatively even across most of the country. But in specific corridors, the Costa Chica of Guerrero and Oaxaca along the Pacific coast, the port city of Veracruz, the cattle ranching interior of Guerrero, it concentrates. And when researchers look closely at exactly where in West Africa that ancestry originated, the answer was not where they expected. And yet, the data said the exact opposite of what the Caribbean comparison would suggest. Research on beta-globin haplotypes, the genetic markers carried in sickle cell trait carriers, in communities of the Costa Chica identified that the enslaved Africans brought to this specific region came primarily from the Bantu-speaking populations of Central Africa, not from Senegambia, not from the Bight of Benin corridor that supplied much of the Caribbean trade. The Costa Chica signal traces primarily to what is now the Central African Republic and surrounding regions. The Caribbean trade and the New Spain trade were drawing from different parts of a continent. The DNA knows which part. Actually, hold on. Before we go further, I want to ask you something, and I genuinely mean this. Drop it in the comments. When you think about your own ancestry, wherever in the world you're from, is there a branch of it that stops? A name you can't trace past a certain generation, a line that just goes silent? Because what the genomics of Mexico is showing us is that the record stops here is not a personal failure. It is the human condition written in bone. Tell me where your record stops, because it might be more connected to what I'm about to say than you'd think. The sickle cell trait is a survival mechanism in high malaria environments. It arrived in the Costa Chica from Central Africa, with the people forced to carry it. Their descendants still show elevated rates of this trait in communities in Guerrero and Oaxaca today. The disease is a genomic timestamp. The genome of illness is also the genome of origin. In 2015, Mexico conducted its first census question specifically designed to count Afro-Mexican identity. 1.38 million people said yes. By 2020, that number had risen to 2.576 million. The undercounting from every previous census, decades in which the category did not exist as an option, mirrors the genomic record exactly. A third stream that was present, contributing, reshaping the population for 4 centuries, systematically uncounted. But the genome doesn't take the census, it simply records the genetic fault lines of colonial Mexico. The asymmetry in who survived, who reproduced, who left descendants, do not describe the past. They map onto the present, and the most precise record of that asymmetry is not in a history book. It is in the chromosomes, active and asymmetric. The Y chromosome does not recombine. It passes from father to son, unchanged except for rare mutations, generation after generation, in a line that reaches from the present into the deepest past. The mitochondrial genome passes from mother to all her children, the same way, the same unbroken line. These 2 chromosomes are the simplest ancestry records in the human genome. And in the mestizo population of Mexico, 130 million people, they tell opposite stories. Researchers analyzing Y chromosome haplogroups across 659 mestizo individuals in 10 populations throughout Mexico found that paternal ancestry, the father's line, was predominantly European, 64.9%. Native American paternal lineages, 30.8%. In northern and western Mexico, European paternal ancestry reaches between 66 and 95% of the sample. The father's line of Mexico is mostly European. Now hold that number next to the autosomal data, the full genome average. The full genome of a mestizo from central Mexico shows roughly 65% indigenous ancestry overall. That number is real. It's confirmed by multiple independent studies. But if the father's line is 30% indigenous, then the mother's line has to be doing most of the work. The mitochondrial record, the maternal inheritance, is overwhelmingly indigenous. This isn't just statistics. This is two chromosomes telling two different stories about the same population. And here is where the data stops being abstract. The mitochondrial record of Mexican mestizos is a record of indigenous women. The Y chromosome record is a record of European men. This is not a description of romance. It is not even primarily a description of relationships. It is a description of demographic consequence, of a colonial encounter recorded at the level of reproductive survival. Indigenous women entered the post-colonial genomic record in far greater numbers than indigenous men. The chromosome frequencies are a measurement. They are measuring something specific. And what they are measuring has a name. It is just not a name that gets spoken in the standard mestizaje story. Sor Juana Inés de la Cruz, born 1648, New Spain, the greatest poet of the colonial Americas. Her father, Spanish. Her maternal line, given everything the mitochondrial frequency data shows about women in New Spain at that time, was almost certainly indigenous. Her brilliance was celebrated in viceregal courts. Her indigenous maternal inheritance appears nowhere in the record of her life. The genome of her country would have shown it. In 130 million people classified as mestizo, the mitochondrial line, the mother's mother's mother, is almost entirely indigenous. The Y chromosome line, the father's father's father, is overwhelmingly European. The genome of Mexico is not a blend. It is a record of who survived what. What they survived, according to the national story, was transformation. And that story deserves to be heard properly before we answer it. José Vasconcelos, philosopher, politician, Mexico's Secretary of Education, publishes La Raza Cósmica, The Cosmic Race. His thesis is not casual. It is one of the most ambitious arguments for human synthesis ever written. That Mexico's future belongs to a people created by the genuine fusion of the world's civilizations, a new kind of human being forged from the encounter of indigenous, European, and African cultures. The mestizo is not a compromise, not a dilution. The mestizo is the achievement. A population that has moved beyond the tribal divisions of the old world into something more complex, more complete. For a century, this framework shaped Mexican education, immigration policy, national identity, and the way Mexico presents itself to the world. And it deserves to be taken seriously on its own terms. Because here is the strongest version of that argument. The autosomal data confirms it. A population that is genuinely 65% indigenous, 31% European, and 4% African, carrying 3 continental genetic streams simultaneously, is not a myth. That population exists. Every study confirms it. The biological reality of admixture is exactly what the synthesis framework predicted. Something genuinely new was created. Something that cannot be reduced to any single ancestor. That is real. The continuity findings from the Villa-Islas study reinforce it further. The indigenous genetic structure of Mexico has not been replaced. It has survived. It threads through the mestizo genome as the dominant component, the foundation held. And the cultural argument runs even deeper. The living presence of 68 distinct indigenous language groups in Mexico today, the persistence of pre-Columbian architectural traditions, agricultural practices, cosmological frameworks. These are not genetic data, but they are evidence of a synthesis that is genuinely two-directional. The mestizo identity absorbed the colonizer as much as the colonized. This is the strongest case. It is genuinely strong. And now here is what the sex chromosome data does to it. A synthesis implies mutual contribution. What the Y chromosome and mitochondrial records show is not mutual contribution. They show directional contribution. The indigenous component flows through the maternal line. The European component flows through the paternal line. Those are not the chromosomes of a synthesis. They are the chromosomes of a specific demographic dynamic, one in which the mixing happened in a particular direction at a particular power ratio that the autosomal average smooths over. The synthesis narrative is not false, but it is incomplete in a way that the genome makes precise. The 65% indigenous autosomal ancestry is real. The question the sex chromosomes forces, whose mothers and whose fathers made that number? And the answer to that question is not found in Vasconcelos. It is found in the frequency of haplogroups. The genome doesn't have a national narrative. It has a record. And the record says, the beautiful idea of the cosmic race was built on top of something the national story preferred not to examine too closely. The data doesn't erase the beauty. It adds a weight to it that cannot be removed. Here is what makes the weight bearable. Not lighter, bearable. The indigenous populations of Mexico were supposed to disappear, not once, multiple times. The megadrought was supposed to replace them with northern nomads. The colonial encounter was supposed to absorb them into a new category. The census, for most of the 20th century, did not count them as a distinct biological heritage. The consumer ancestry test compresses them into one number. The national synthesis narrative folds them into the foundation and builds something else on top, and they are still there every time. The ancient genome data from the Sierra Gorda shows continuous occupation through 2 centuries of catastrophic drought. The mitochondrial data from the mestizo population shows the maternal line intact, dominant, threading through 130 million genomes. The Garcia-Ortiz dataset shows 60 distinct ethnic groups maintaining enough internal genetic structure to be differentiated at the population level, meaning the mixing happened, the admixture happened, and the internal diversity survived anyway. These populations were not preserved in amber. They mixed, and they remained distinct. This is the finding that the standard model of Mexican genomics genuinely did not predict. The assumption was that admixture at this scale, 500 years of population mixing across a continent, would homogenize the genetic structure of the indigenous component beyond recovery, that the internal diversity of pre-Hispanic Mexico would blur into a single reference cluster. It didn't. Moreno Estrada's 2014 study found that the genetic stratification of Mexico's indigenous groups maps onto pre-Columbian population structure. The ancient divisions are still there. The autosomal diversity of mestizo populations correlates with the specific indigenous groups nearest the geographic origin of each mestizo community. The past is not erased, it is embedded. The wrong villain in the standard collapse narrative is not colonialism. That is precisely documented. The wrong villain is time. The assumption that time erases. The genome of Mexico says time records. It does not erase, it layers. Every drought, every conquest, every population movement, every century of admixture, all of it exists simultaneously in the same living genome, waiting for someone to sequence it and ask the right question. What the field has not yet answered, and what the Villa-Islas study specifically names as an open problem, is who the mestizo populations nearest to the Cañada de la Virgen and Sierra Tarahumara sites carry in their genomes today. The ghost signal was identified in ancient individuals. Its modern distribution has not been fully mapped. There are living people in Guanajuato and Chihuahua who may carry UpopA or UpopA2 at detectable levels. No study has looked yet. Here is the thing about a ghost population. The word ghost in genomics is a technical term. It means a population whose existence is inferred from the genetic signal they left in other people, but for whom no direct ancient sample has been recovered. We know they existed because their DNA is in someone else's bones. We know their approximate ancestry contribution. We can model their rough position on the human family tree. We know almost nothing else. No name, no language, no burial site that belongs to them. No archaeological culture we can point to and say, that was them. The bones at Cañada de la Virgen are not their bones. They are the bones of people who carried their signal, the way you might carry a piece of furniture from a house that no longer exists. The furniture is real. The house is gone. We have never found the house. UpopA2 contributed 17% of the ancestry of the ancient individuals at Cañada de la Virgen. That is not a trace amount. That is not background noise. That is a substantial genetic contribution from a real, sizable population that lived in the Americas, probably for thousands of years, and left no other record. The only way to find them now is to sequence more ancient individuals from more sites across Mexico, and hope that somewhere, in a burial that hasn't been excavated yet, their genome appears at higher concentration. Maybe that skeleton exists. Maybe it has already been found but not sequenced. Maybe it is still in the ground. This is where you are now, watching this video in 2025. The science is mid-sentence. UpopA, the Mixe ghost signal, has a slightly different situation. It exists in a living population, which means it can be studied. Which means its position in the human tree can be refined as more reference populations are added to the global databases. Researchers are now asking whether UpopA represents a lineage that diverged from the ancestors of other Native Americans before the main peopling of the Americas, a branch that split early and moved separately. Early data points in that direction. The field hasn't settled it. If that interpretation holds, the Mixe of Oaxaca carry a genetic lineage that diverged from the main stem of Native American ancestry before most the people who would become Native American had even crossed into the continent. They carry the oldest American branch. They have been carrying it in relative isolation for longer than most civilizations have existed, and they have no way to trace it further than the science currently allows. Now think about your own genome for a moment. If you have any ancestry from Mexico, from any region, any community, any generation removed, there is a non-trivial probability that somewhere in your mitochondrial record, in the deepest maternal branch, there is a woman who carried a lineage that traces not to the named civilizations, not to the Aztec or the Maya or the Zapotec, but to a people with no name in any language currently spoken. A people who lived, built something, mixed with their neighbors, contributed their biology to the human story of an entire country, and left no word. What the genome of Mexico keeps returning to, past the admixture percentages, past the colonial demographics, past the ghost populations, is a single unresolvable fact. The most important people in the biological history of this country are the ones we cannot name. The ancient individuals at Cañada de la Virgen did not know who their ghost ancestors were either. The signal was already old when it arrived in their bones. It has been passing forward ever since. The science will sequence more individuals. The reference panels will expand. The ghost populations may eventually be found. A burial somewhere in the Mexican high desert with the signal at full concentration. The house instead of just the furniture. That study has not been published. That skeleton may not have been found. The field is still looking. Until then, what the genome of Mexico holds is not a mystery waiting to be solved. It is a fact waiting to be named, and the name does not exist yet.